Hello and welcome back to Science of Medicine. Today I want to talk about the immune system. This is a vitally important system that keeps us alive by virtue of the fact that we're constantly being attacked by various pathogens. Most of these are microscopic pathogens as opposed to animals or plants that are trying to kill us. But nonetheless, they still can be deadly, and death by various infectious causes is still a major cause of mortality throughout the world. So the way that our bodies have adapted to create this defense system have two big components. The first are what are called the innate defenses. These are defenses that don't discriminate based on what kind of a pathogen it is. It could be a virus, it could be a bacterium, it could be a fungus. They're things that equally protect across those different pathogens. And the other is an adaptive immune response. The adaptive immune response is much more like a sniper kind of thing, something that's going out and looking for a particular pathogen and then trying to kill that. Under the innate things we have are barriers like skin and mucosa in our gastrointestinal tract. We also have in our respiratory tract ciliated cells that have these little hair-like follicles that basically move pathogens that are trapped in mucus up so that we can cough them out or swallow them. There are physiologic barriers. For example, in the stomach, the hydrochloric acid gives a very low pH that kills most pathogens. We can adjust the temperature of our bodies to retard growth of potential pathogens. There are a sequence of proteins that are collectively called complement that we'll talk about in a minute that basically have different pathways that lead to the destruction of pathogens. There are certain types of phagocytic cells that go around and eat invading pathogens. And of course, if I damage my body in some capacity, if I cut my hand or something along those lines, I will have inflammation or a way of trying to wall off from potential pathogens entry into my body. Under the adaptive portion of the immune system, those are mostly just held within the B and T cells, and we'll talk about those in detail. So I need to give you some basic understanding of some terminology that is used throughout the discussion of the immune system. The first is the idea of an antigen, and I alluded to this and talked about this when we were talking about the ABO blood types. An antigen is just a term that we use to describe a portion of a molecule, in this case on a pathogen, that is able to induce an immune response. Now, proteins are very good that do this, but polysaccharides also will do it. The two of the new COVID-19 vaccines that are used use messenger RNA as that antigen. There are also immune components that interact with those antigens. Those are things like antibodies, which are made by B cells. There are 5 different classes, which we'll talk about. And then also antigen-presenting cells, not only B cells, but phagocytic cells and dendritic cells that basically will phagocytose or consume a pathogen, chew it up into small pieces, and put that into what's called an MHC or major histocompatibility complex and present it to T cells. 1,000 years ago, or more than 1,000 years ago, when invaders would try to take over a castle or something, if they were defeated, the heads of those warriors would be put up on a spike outside of the castle gates to thwart any other invaders. Kind of like that in these antigen-presenting cells. It is basically saying, look, this is a potential pathogen to us. Go out and look for this. So I mentioned to you, as part of the innate immune system, is this idea of complement. Now, complement is a series of proteins. Glycoproteins are synthesized by hepatocytes in the liver, and they ultimately lead to the lysis of cells. Those cells can be bacteria, They could also be viruses. It's non-specific. By coating these pathogens also with different proteins, it supports what's called opsonization. Opsonization is the process of phagocytosis that I had mentioned earlier, and then helps with the clearance of that pathogen through the adaptive immune system. It also facilitates their removal in the spleen, and it binds to receptors on certain cells and serves as an augmentator of the immune response. And I'll show you that in just a second. So this is a busy picture. There are several pathways, the classical and the alternative pathways, that produce a number of effects. So as you can see here, if I have under the classical pathway a microbe, or under the alternative pathway, various proteins, they're listed as complement, and they go complement 1, 4, and there's binding. For this purpose, and this is a beautiful process, I'm not going to talk about the details of which binds with which, but what I want you to see is that the binding causes various reactions, and you get components that come off, like this 3A component. These are sometimes generically referred to as cytokines. They have other effects that lead to inflammation, that lead to the recruitment of other immune cells to the location. And that's part of how they promote this chemotaxis and this phagocytosis. Ultimately, I'm going to try to insert pore into the pathogen, into the microbe, that will lead to its demise, but I'm going to do so much more in this process by creating these other molecules and recruiting other immune cells. Now let's talk a little bit about those immune cells. They arise from stem cells, and if I look at those stem cells, I notice that they sort of branch in two ways. They can branch off into a myeloid cell line or a lymphoid cell line. The myeloid cells that generate from that are part of that innate immune response, but the lymphoid are those that are going to be part of that adaptive immune response. They're going to give rise to natural killer T cells, and B cells, whereas the myeloids are going to give rise to a whole host of cells. The erythrocytes that we talked about in the hematology section, platelets that are actually going to be used as part of the patchwork if I have blood vessel compromise and I'm starting to leak out fluid, but also mast cells, myeloblasts, which lead to basophils, neutrophils, eosinophils, and then ultimately cells that lead to macrophages. So I want you to think about these stem cells giving rise to myeloid and lymphoid, the lymphoid becoming part of your adaptive immune response, the myeloid part of the innate, but the myeloid being much more varied in terms of what they actually produce. So let's talk about some of the detail. As I mentioned to you in the lymphoid side, being part of the adaptive immune response, the B cells are going to produce a class of molecules called antibodies. And the antibodies are going to have several effects, which we'll talk about in a little bit. T cells are going to be also involved in the direct killing of pathogens, but also they're part of your immune modulation. That is to tamp down the immune system when it doesn't need to be activated, but also to ramp it up when it does need. And natural killer T cells also are part of this pathogen protection by killing specifically identified cells. On the myeloid side, we talked about the erythrocytes and the platelets when we talked about hematology. Let me talk about the immune components there. There are neutrophils. Neutrophils are a very abundant class of myeloid progeny that actually are involved in phagocytosis. Once they're phagocytosed, peroxisomes will lead to degradation of those components of the material that has been phagocytosed. Monocytes, which float around in the blood, become macrophages when they go into various areas outside the blood vessels. They are a type of antigen-presenting cells. They will go and phagocytose a pathogen and then present in this MHC complex to other immune cells portions of that pathogen, that is antigens, to allow for the ramping up of immune response. Now, macrophages go by lots of different names, depends upon the location. They're in the liver called Kupffer cells, histiocytes in the skin, alveolar macrophages in the lungs. They go by different names based on where they are, but they're all still macrophages, and they started out as these myeloid cells that became monocytes floating around in the blood vessels. Eosinophils are very specific to fight off parasitic infections. So if you have parasitic worms that are endemic in your area, the eosinophils are going to actually lead the charge to try and off those pathogens. Mast cells, such as basophils, are histamine releasers. They promote inflammation and thereby wall off and give the body time to actually go and fight off this pathogen. Now, one of the things that's very important for any immune system is that you have to determine whether whatever you're encountering as a cell is of the body or foreign. Because it's not like, you know, all the pathogens wear red shirts and everybody knows that's a bad thing, or blue shirts or yellow shirts or something like that, and that's okay or pathogen. So what we have are a sequence of protectors. The first thing that we have is that most all of the cells in your body will have a proteoglycan, which is an MHC, major histocompatibility complex, type 1, that presents itself, it's present on almost all of your cells, that says, I'm part of your body. Do not initiate an immune response. There are some exceptions. For example, sperm cells don't have this, so that they don't actually get, they're not attacked within your body. They're in a sequestered area. But nonetheless, these MHC type 1 that are expressed on nearly all of my cells are going to want to tell the body that this is self. How do you let the immune system know that these are not supposed to be attacked? And the way that you do that is in the maturation of those cells. T cells in particular will go through and do this. And what they do is, if they actually bind too tightly to the MHC type 1 complex, they get eliminated as part of your immune system. Obviously, there are a number of autoimmune disorders, things like lupus or rheumatoid arthritis, where this goes awry, and you think that your body is being invaded by something foreign, when in fact it is not. Now, there's also an MHC type 2, which I've alluded to earlier. This is a protein that will be the place where you present foreign antigens by these antigen-presenting cells, dendritic cells, B cells, macrophages. So any of those will engulf this pathogen and present portions of it, generally fairly small portions, to T cells via this MHC type 2 complex. And that way it says, look, this is a foreign invader, go seek it out and kill it. I show this schematically here, where I have a T cell interacting with antigen-presenting cells, in this case a macrophage, in this case a B cell. And you see that there's a portion of that pathogen, this little red squiggly line here, that's been presented to this T cell, which will allow it to secrete a molecule, and then in this case stimulate the B cell. This is a paracrine type of interaction. Or in the case of the macrophage, simply the macrophage releasing a stimulatory agent and a small molecule that will bind with the T cell receptor to allow for it to become activated. All right, so let's talk about the adaptive immune response. So as I mentioned to you, that's made up of your B and your T cells. B cells produce antibodies that are to specific antigens. We learn those over our lifetime. They are made in the bone marrow, but also mature in the bone marrow. So they are B cells. You can think of them as being maturing in the bone marrow. And they secrete cytokines also that are responsible for immune system modulation and stimulation. But they produce antibodies. That's their most prevalent kind of role in the immune system. And they actually form what are called plasma cells, which just crank out these antibodies. And there are several types of antibody classes that there are. IgM, IgG, IgA, IgE, and IgD. And they serve different roles. IgM, which is the first antibody class that is made after a new pathogen invades your body, normally ramp up within 1 to 2 weeks, which is why many diseases that you haven't seen before, it normally takes you about a week or two to get over them. Now, the IgM molecules will then convert into IgG molecules, antibodies. And these are the antibodies to antigens that have been exposed to your body before. It could be from a vaccination, it can be from prior disease. And then that allows for a faster memory response. And by that I mean that when your body encounters some pathogen that it's seen before, you don't have to go through the IgM stage, which takes you about a week or two to ramp up the response, but instead the IgG can actually ramp up very rapidly, and it allows for a much faster response once you have primed that system. IgG are also antibodies that can cross the placental barrier, so they can be transmitted from mom to the fetus and afford some protection. IgA are a class of antibodies that are present in secretory things, such as breast milk, the gastrointestinal tract, the pulmonary tract, the respiratory tract, the urinary tract. And it's mostly designed to trap antigens so that they don't actually get into your body. IgE are responsible for mediating hypersensitivity reactions. These are allergic reactions in particular. And IgD, we don't know the details of exactly what it does and how it does it, but we know that it is important in B cell activation. So let's talk a little bit more about antibodies. Antibodies are simply these proteoglycans that are made up of different components. We have in this protein backbone a constant region, which is called the Fc region, and variable regions. It's the variable regions that are going to change based on the antigen that is presented to them. And it's a brilliant, wonderful way that actually we make these adaptations. And I would encourage you, if you have time, to read about the immunology of this, because it's absolutely fascinating how we go through these changes and develop this. But suffice it to say for this, that you do actually form a unique cleft, a unique area that will interact ideally with just one antigen at these antigen binding sites. And then once you've done that, if you have a lot of these antibodies that are produced, that will lead to this things like opsonization or cell dependent or cell mediated toxicity, where you will actually kill that invading pathogen. Now the T cells, also part of that adaptive immune response, are necessary for doing this and having the B cells work their magic through antibodies. They are made in the bone marrow also, but they're called T cells, and that's because they mature in the thymus. The thymus is a gland that's up here at the top part, just below your neck, that actually is really critical for the maturation of these T cells, because they have to be able to bind to antigens presented to them in the MHC2 receptors, but they also can't bind and cause an immune response to the MHC1 receptors, because that's part of your body. They need to recognize that as your body. You've heard a lot of these T cells, CD4, CD8, for example, that are there. And the T cells are responsible for several things. First off, they directly destroy infected cells. They can induce what's called apoptosis, or programmed cell death. They also will secrete cytokines to stimulate the immune system, for example, for B cells to ramp up their production of antibodies. A lot of these cytokines then serve other purposes. For example, interleukin-1 or IL-1 is associated with increasing temperature There are others that are associated with inflammation. So much like some of those complement fragments that come off, you also have specific cytokines that are stimulated by T cells to help with this immune response. One of the things that is necessary for this system to work is you have to bring the pathogens into proximity to the cells that are going to kill the pathogens. So part of the adaptive immune response. And that is done in lymph nodes and in the spleen, which is a modified type of lymph node. So all throughout your body, you have these nodal structures where you have lymph, which is a fluid that's going to be taken from your blood vessels that leak out a little bit. We'll talk about that in the cardiovascular system. And it will bring in with it these antigen-presenting cells as well as pathogens into this structure that is a lymph node. And it will meander through these various B and T cells that will then allow them to interact and say, hey, have you seen this pathogen? No, you haven't. Let me go over to this other B cell. Have you ever seen this pathogen? And it goes through that process and then drains it out, and then that goes back eventually into your bloodstream. If, in fact, there has been an antigen that has been present before, then that area of the lymph node starts to propagate, and you start to generate more and more of those B cells to ramp up, which is why when you have an infection, you will end up having swollen lymph nodes many times. So we think about those as being, let's say, up in your neck if you have some sort of respiratory issue, because that's one of the first places that it will go. And that will help with this process of getting immune cells that are in the adaptive immune response into contact with the antigens that are actually going to stimulate those. Now the spleen, which as I mentioned to you before, is located in the left-hand portion of your abdominal cavity, also has this similar structure. We talked about this when we talked about the erythrocytes. But it has this white pulp area, which serves the same purpose as a lymph node. It allows for basically these immune cells to interact with potential pathogens, and then allow for the activation of that portion of the immune system. So I hope that overview has helped a little bit. Let me see if I can test that. So let's think about this application. The Norwalk virus is a causative agent for a lot of types of viral gastroenteritis. You have some vomiting, some nausea, some diarrhea. Which of the following would be an example of the body's adaptive response to this kind of a virus? Low pH in the stomach, inflammation of the mucosal surface of the GI tract, secretion of IgA in the GI tract, fever. So think about that for a second. Well, I hope that you recognize that the key issue here was that while all four of these things can protect you from this pathogen, there's only one in the adaptive response, which is the secretion of IgA, which is coming from B cells, that is actually in this part. And that's really important that you distinguish between this innate and adaptive types. Well, I hope that that has been helpful for you in this introduction to the immune system. I will talk more about medical physiology as we go through these various snippets. As always, if you're interested in these subjects, please feel free to leave me a like on this or to subscribe to this channel, and I'll see you soon.